lecture down into like 3 parts. We talked about glycolysis, we talked about the TCA cycle, and we've made limited amounts of ATP between these two. The third part is following the electrons through the so-called electron transport chain and the process of making ATP by oxidative phosphorylation. So in this first step with glycolysis, we get just a pair of electrons coming out at that one step. We get 2 NADHs from each glucose there. In the pyruvate dehydrogenase complex, we get another couple NADHs. As we go into the citric acid cycle, well, there are 4 places where electron pairs are donated into this pool of NAD and NADH and FAD, these reduced carriers. So all the way along, we're collecting electrons off the glucose and its breakdown products. And the electrons always go to either FAD or NAD. And then in the last step of this process, those electrons are donated to the electron transfer chain that's incorporated into these membranes, these internal membranes, the electron transport chain. It's all throughout those inner membrane of the mitochondria. They funnel those electrons to oxygen with the electrons reducing the oxygen, the molecular oxygen gets reduced to water. This is energetically downhill, this flow of electrons. So much downhill that you can generate lots of ATP from the process. So this kind of puts it all together. Glycolysis, the pyruvate dehydrogenase reaction, the Krebs cycle, all of these processes putting electrons into this pool of reduced carriers. And then in the last step, we harvest the energy of those electrons to generate ATP. So these 2 steps, the moving of the electrons and the making of the ATP, that's what oxidative phosphorylation is. It's a process of making ATP. Those 2 go together. And as we'll see, an integral part of this process of converting the electron flow to ATP synthesis is the pumping of a simple thing, the movement of protons across the gradient. To create a gradient of acidity or a proton gradient across this membrane. So we will be, in essence, taking protons from the inside in the matrix, pumping them out here, creating a very acidic environment here, and this gradient will be used to drive ATP synthesis. We'll get into the details of that shortly.